Welcome to the Deep Dive, your shortcut to being genuinely well-informed.
Hey there.
Today, we're tackling something that honestly sounds like it's straight out of science fiction.
Mhm.
Bringing an animal back from extinction, we're talking about the Tasmanian tiger, the thylacine.
Yeah, Australia's vanished apex predator.
Oh.
Iconic, really. Last one died back in 1936. It's become this symbol of, regret.
Exactly, and for almost a century, extinction just meant gone forever, a one-way street.
Right. But that whole idea is being, well, seriously challenged now.
Mm.
There's this incredibly audacious project underway.
Colossal Biosciences and the University of Melbourne's Tiger Lab.
That's them. They're aiming to de-extinct the thylacine. Bring it back. It's a massive undertaking. Redefines things.
So our mission today is to unpack the, frankly, mind-bending science behind this.
Yeah, look at who's involved.
And get into the really big ethical and ecological questions this whole thing raises.
Definitely.
So get ready. We're exploring how they're trying to do it, and maybe more importantly, what it all means. Let's kick things off right at the start. The blueprint. The thylacine's DNA. How do you even get that?
Well, every big comeback story starts somewhere small.
Right.
A kind of whisper from the past.
Yeah.
For the thylacine, that whisper was locked away for over 100 years. Scientists, Professor Andrew Pask leading the charge here, did some amazing genetic detective work.
Okay.
They managed to sequence a thylacine genome that's, well, it's near perfect. They got it to 99.9% accuracy by 2024.
Wow, 99.9% from what?
From a museum specimen, 108 years old.
Incredible.
And the really key thing, they extracted RNA from let's just say a putrid preserved thylacine head.
Oh, wow. Okay, RNA. Why is that so important?
Because RNA tells you about gene expression, which genes were actually switched on, doing things. It gives you that layer of genetic authenticity you absolutely need.
Got it. So you have this super detailed genetic map, but you can't just print a thylacine from that sequence, can you? What's the next piece?
No, exactly. You need a living template, a sort of biological starting point.
Right.
And they found one, the fat-tailed Dunnart. It's this tiny mouse-like marsupial.
A dunnart. Why that specific animal? Seems like a big size difference.
It is, but it's the closest living relative they could work with. And crucially, it has a very short gestation period that makes it much, much better for this kind of experimental reproduction work. Faster cycles.
Okay, makes sense.
Okay.
So dunnart is the template. Now, the genetic wizardry, CRISPR. We hear about CRISPR all the time. How are they using it here?
Right. So they have the thylacine genome map, and they have the Dunnart genome map. They use CRISPR-Cas9. That's the gene editing tool.
The molecular scissors?
Kind of, yeah.
No.
To precisely snip the Dunnart DNA and insert key thylacine genetic sequences.
So editing the dunnart cells.
Exactly.
Yep.
Modifying the genes that control things like size, skull shape, even trying to get those famous stripes back.
Stripes? Yeah.
Professor Pask is pretty confident they can recreate those major physical traits, and they've got expertise from people like George Church involved, too, a big name in genetics.
Okay, so they're engineering these thylacine-like cells in a dish, got the blueprint dialed in. What's the next big step to actually get an embryo? Is this cloning like Dolly the sheep?
Yes, essentially. The next stage involves what's called somatic cell nuclear transfer, or SCNT. Same basic idea as Dolly.
Okay.
You take the nucleus, the genetic core, from one of those engineered thylacine-like cells. Then you put it into a Dunnart egg cell, where the original nucleus has been removed.
And that tricks the egg into developing?
Theoretically, yes.
Okay.
It should start dividing and developing as if it were a normally fertilized egg, but carrying that engineered thylacine DNA, a cloned thylacine embryo.
Right, but here's where my mind kind of boggles. Marsupial reproduction is different, isn't it? Tiny babies, pouches. How does a tiny Dunnart possibly carry a thylacine embryo, even if it starts small? That seems like a huge hurdle.
It was a colossal hurdle, absolutely.
Yeah.
But this is where they announced a really massive breakthrough just in 2024. So the team figured out how to induce ovulation in Dunnarts, getting them ready to be surrogates. But the real game changer, they successfully grew fertilized embryos to mid-gestation.
Mid-gestation, like halfway.
Yeah, roughly halfway through the pregnancy. And they did it outside the body in an artificial uterus device.
Whoa, an artificial womb for marsupials.
Exactly. It's a world first for marsupial reproductive tech. It basically bypasses a huge part of that biological mismatch problem.
So they grow it partway in the lab device.
And then transfer it into a surrogate Dunnart's womb for the final few weeks of gestation.
That is genuinely astounding progress. But okay, even then, You end up with a tiny, like, bean-sized thylacine joey, right?
Right. Still very underdeveloped compared to placental mammals.
And a Dunnart pouch isn't exactly built for a growing thylacine. What's the plan for that, the final stretch?
You've hit on the next big challenge, neonatal care. It's the ultimate hurdle.
What are they thinking?
They're exploring some really innovative ideas. Developing specialized artificial pouches is one possibility.
Like a little high-tech incubator pouch?
Sort of, yeah. Or maybe even fostering the joeys with other, perhaps larger marsupial species. It's still being worked out.
Okay. So with all these breakthroughs, what's the timeline looking like? When might we actually see something?
Professor Pask is optimistic, but also, cautious. He's estimated that a thylacine-looking baby.
Yeah, thylacine looking. Interesting phrasing.
Well, yeah. He thinks that could potentially be born within the next 3 to 5 years. So
Yeah.
maybe 2027, 2029, somewhere in that ballpark.
3 to 5 years. That feels incredibly soon for something this ambitious.
He does. Which leads us to the funding, because science like this isn't cheap.
Right. Who's bankrolling this moonshot?
The major driver is Colossal Biosciences. They've put in something like $10, $15 million specifically for the Thylacine project.
That's serious money.
It is. And their whole model is about attracting new investment, often from high-profile sources. You hear names like the Hemsworth brothers, Paris Hilton even.
Really? Hemsworth and Paris Hilton.
Yep. That kind of interest brings in capital that, frankly, traditional science grants just wouldn't cover. It lets them move much faster. It's a different way of funding these big ideas.
Okay, which brings us inevitably to the big why question. Why do this? Is it just fixing a past mistake, or is there something more? Because you always hear the argument, shouldn't that money go to species that are endangered now?
And that's a totally valid point. It forces a really interesting conversation about what conservation even is in the 21st century.
Mm.
So, on one side, you have the moral argument, humans wiped out the thylacine, period. So maybe we have an obligation to try and undo that damage.
A moral debt.
Kind of. Professor Pass points out, we play God all the time with things like IVF or genetic interventions in farming. He sees this as an extension of that, perhaps.
Okay, but what about the counter argument, the resources for living species?
Right. But the counter to that is that this project isn't necessarily taking money away from existing conservation. It's bringing new money in.
Ah, attracting different kinds of investors.
Exactly. And maybe even more importantly, it's driving the development of technologies advanced CRISPR for marsupials, these new assisted reproduction techniques.
The artificial womb tech.
Yeah. Those tools could be game changers for living endangered species, too. Imagine using CRISPR to give Tasmanian devils resistance to their facial tumor disease.
Right, or helping quolls deal with poisonous cane toads.
Precisely. So the tech developed for the thylacine could end up helping safeguard lots of other species. It's a potential toolkit for biodiversity.
So it's potentially a win-win tech-wise. What about the ecological side? If they succeed, what's the vision for a return to thylacine in Tasmania?
Ecologically, the thylacine was the apex predator. Its removal caused what ecologists call trophic downgrading. Basically, the ecosystem got knocked out of balance.
How so?
Well, without the main predator, maybe herbivores become overpopulated, or maybe smaller predators like feral cats run rampant.
Feral cats are a huge problem in Australia, right?
Massive. They devastate native wildlife. So the idea is that a returned thylacine could potentially help control herbivore numbers, maybe suppress those invasive feral cats, and restore some of that natural balance. It's
Yeah.
about putting a key piece back into the ecological puzzle.
That's a compelling vision. But it's been, 90 years? Do the native animals in Tasmania even remember the thylacine? Is there still that instinctual fear, the ghost of predators past?
That is an excellent and critical question, because just dropping a predator back in isn't enough.
Right.
And Pask's team is actually looking into that right now. They're doing experiments, testing how animals like wallabies react to life-size thylacine models.
Seriously, they're showing wallabies thylacine models.
Yeah, to see if those innate anti-predator responses are still there after all this time. It's fascinating, and it's crucial for planning any potential reintroduction down the line.
Yeah.
That whole predator-prey dance needs time to reestablish.
It really does. Okay, so summing up, this deep dive shows just incredible scientific progress from getting that near-perfect genome
Yeah, from a century-old sample.
to growing embryos in an artificial womb. The leaps are just undeniable.
Absolutely staggering, technically. But it does raise a fundamental question, one for you listening. If or when a thylacine baby is born,
Yeah.
is it really a true thylacine?
Or is it more like a thylacine-like animal engineered from a Dunnart template?
Right. Because an organism is shaped by so much more than just its DNA sequence,
Yeah.
environment, epigenetics. It's complex.
Definitely something to think about. And maybe here's a final thought to leave you with. If science can start to chip away at the idea that extinction is forever, potentially bringing back the Tasmanian tiger, what does that truly mean for how we view nature?
Mm.
How could this kind of groundbreaking science be used responsibly, thoughtfully, not just to resurrect a few lost species, but maybe to help preserve all biodiversity? How does it change conservation itself?
Yeah. The journey, it feels like it's really only just beginning.